this afternoon is Josh Berke, who's currently director of the addiction program at the University of California, San Francisco. He did his undergraduate work at Trinity College, Cambridge, did his graduate school at Harvard and a postdoc in Boston, working with Howard Eichenbaum. He started his own lab in biopsychology at the University of Michigan in Ann Arbor. And then in 2016, we had the good fortune to attract him to UCSF, where he's currently the Rudy Schmidt Distinguished Professor of Neurology and Psychiatry and Director of the Wheeler Center for the Neurobiology of Addiction. Josh?
Hi, can you hear me okay?
Yeah.
All right. So I can't, for some reason I've got it, so I can't see anybody to see my slides, but that's okay. So thank you very much.
Okay.
Behind me?
Yeah, the light
okay?
behind you is kind of obscuring your face.
That's my glow.
Right.
Well, I'm going to go on unless it's too much of a problem.
Okay.
So thank you very much to the organizers for inviting me to talk. It's been a fantastic conference, at least until now. I'm going to, first of all, thank the people who did the work in my lab. Some wonderful people, including Ali Mohabi, shown here when he still looked respectable, Arif Hamid, and Jeff Pettibone. And the story I'm going to tell in some ways starts in the 1950s. So with these amazing experiments by Olds and colleagues, where they showed that there were places in the brain that you could tickle with electrical current. And if you hook them up to a lever, then rats would learn to push that lever a lot. They would be highly motivated for this intracranial self-stimulation. And there was a great deal of mapping of where does this happen in the brain. And even though there are a number of places, the most potent places in the brain are clustered along the mesolimbic dopamine pathway. And this is a dopamine conference, so I don't need to introduce dopamine too much, other than to say that for this talk, I'm going to be talking about this mesolimbic pathway from the ventral tegmental area to the accumbens. So this has often been referred to as a reward pathway, but what exactly does that mean? So in this nice review a number of years ago by Kent Berridge and Terry Robinson, they showed that they described how you could break up this concept of reward into some more specific concepts. One of those ideas of reward is the pleasure, the hedonics, the liking of rewards. A second concept is this idea of reinforcement, of learning from rewards. And a third has to do with motivation or wanting rewards, driving you forward to obtain more rewards. And we've known that dopamine is involved in many reward-related processes, but not all. So for example, liking doesn't seem to be involved, doesn't seem to strongly depend on dopamine. So if you take an animal, so first of all, there are these highly conserved facial expressions when mammals lick delicious sweet things. And if you totally remove the dopamine from the striatum and you drop sweet sucrose liquid into a rodent's mouth, it'll still show these licking reactions as if it still likes the sugar. But what it won't do is work to get that sugar. So if the sugar is right there, it'll consume it. But if it has to move even a few inches to get it, it won't get it. It'll starve to death. So this and many other kinds of experiments showed some relationship between dopamine and motivation. And of course, we also have a tremendous amount of evidence that dopamine is involved in this learning aspect of reward as well. And we've already heard many times in this conference about the very seminal work of Wolfram Schultz. And the point I'm trying to make here is simply that a delicious fruit juice that initially provokes a dopamine cell firing response doesn't later on, even though the juice is still delicious and still liked. All right. So this is, as we've heard many times now, the concept of reward prediction errors. And I just wanted to sort of highlight that part of the reason why these views are so influential in the field is because of this really nice connection between these sort of systems level observations and these cellular level observations. So here's a very nice slide showing work from Kasai, Yagashita, and colleagues, showing that if you have this conjunction, you take a ventral striatal D1 neuron and you stimulate it at the same time as you stimulate a specific presynaptic terminal, and you give dopamine at just the right time, you can cause that spine to grow before your very eyes. So this shows that dopamine seems to serve this role as a modulator of plasticity on these timescales. Okay, so if we think about this influential reinforcement learning framework, as we've already heard in this panel, there's the basic, to strip things down, at least in this ventral striatal circuit, we often think about these values. These are reward predictions. And in this sort of circuit, this interplay with reward prediction errors. And the point of reward predictions is to drive you forward, to motivate you in order to obtain rewards, right? And the point, and that's the motivation side of things. And the point of reward prediction errors is to serve as a learning signal. So the point of an RPE is to achieve plasticity in order to update your values and so you have the right values, right? It doesn't do anything else, at least in these RL models. Okay. So one important thing to keep in mind, as you listen to my talk and to other talks, other amazing, wonderful talks in this conference, is what should RPEs do, right? So as a situation becomes increasingly well predicted, what you want is your error signals to diminish and go away. It's important that they should go away because otherwise they may continue to evoke plasticity, which is not what you want if your predictions are already accurate. All right, so there's going to be 3 themes in this talk. I hope I get through them. So one is this idea of motivation versus learning that I've already introduced. And manipulations show that dopamine is critically involved in both motivation and learning. But how do we see this, or how do we observe this in dopamine dynamics? The second is this idea of tonic versus phasic. These have been very influential concepts for many decades now in the dopamine field, but are they real concepts? Are there really distinct slow and fast timescales for dopamine signals? And the third is going to be firing versus release. So can we fully account for forebrain dopamine signals by examining midbrain dopamine cell spiking? And if we can't, what else is going on? So these issues of how to reconcile the learning and motivational aspects of dopamine have, I think, been understudied, but they haven't been completely ignored. And here's a review paper by Wolfram Schultz from a number of years ago, essentially proposing that dopamine achieves these different functions by dynamics on different timescales. So maybe it's the case that fast phasic bursts of dopamine cell firing encode reward prediction errors to do learning, and that there are these slower changes over here maybe that do motivation. That would be tonic dopamine. I think it's also worth pointing out in passing that it's very rare when people talk about tonic dopamine to actually define a timescale that they're talking about. I hope we can change that. But usually it's often defined just as whatever the timescale your microdialysis probe is sampling dopamine. So there have been some more specific computational proposals. Here's a nice one from Yale and colleagues a number of years ago. And they suggested there was a separate tonic dopamine signal that was encoding motivation. And specifically, if you can keep track of the rate at which you're getting rewarded for some activity, that's a very useful thing to do because it can help you decide whether engaging in that task is worthwhile. It's a good use of your time, or you should be using your time to do something else. All right, so these are some of the background ideas and concepts that we were thinking about when we started doing some of these dopamine experiments a few years ago. And at this point, I need to introduce the behavioral task that we're working with. And so this is a rat in a box. And what happens simply is that one of these lights comes on to begin the trial, and the rat sticks his nose in the hole. And I'll show you that here now. So the trial starts, light comes on, and then the rat sticks his nose in. And there's some delay. The rat takes a variable amount of time to go stick his nose in the hole. And we're going to call that the latency. And I just want to say that even though there are other events in this task, it's not, this is the key thing for most of this talk, right? So it's not inherently a complicated situation. All right. So after, I'm getting these flashing things. After the rat sticks its nose in the hole, it waits for some variable amount of time until it hears a go cue. It's just a white noise burst. And at that point, it pulls out its nose and sticks its nose in the side port, and it can choose either left or right. And sometimes as it sticks its nose in the port, it hears a click that is the sound of the sugar pellet being delivered behind it, which it can then go and collect. And it's a probabilistically rewarded task. So and these probabilities change every now and then. So this is just a schematic. So tall ticks mean a reward, and short ticks mean no reward. So in this series of trials, the rat tried left, got a reward, tried left again, no reward, left again, no reward, try right, no reward, and so forth. And a full sequence of a full session might look something like this: a few hundred trials, varying probabilities without telling the rat, of course. And I hope you can see that in the smooth time course here, that the rat is adapting so that when left is the better choice, it mostly chooses left, and when right is the better choice, it mostly chooses right. So the rat is adapting its choices, but not just its choices. It's also adapting its motivation to play this game. So if we ignore whether actions were left or right and just look at reward density, you can see there's some times when the rat's getting lots of rewards and sometimes when he's getting fewer rewards. And the blue line here on an inverted scale is just the latency. How eager, how quick is the rat to stick its nose in the hole once the light comes on? And these are these two aspects of adaptive behavior in this task. So one is that as you transition into a block where left is better, then the rat progressively switches over to choosing left and vice versa for left. And this is this adaptive motivation aspect. So when the rat has gotten lots of rewards recently, all 10 of the last 10 trials have been rewarded, it's super quick to go stick its nose in the hole once the light comes on. But if it hasn't got any rewards for a long time, then it says, why should I bother? And it takes a long time. Okay. So how does this latency dependence on reward history come about? And it turns out that if you plot the timing of nose entry, and then what happens is that when the rat's received lots of rewards recently, it's more likely to make a decision to initiate the approach to that light, all right? So that's shown as a hazard rate here. So really what's going on here is a reward history is modulating this something like a decision threshold, a decision that a certain activity is worthwhile. Okay, so now we have this task, and I've explained these sort of motivational and learning aspects so we can study dopamine in this task. And I'm going to start by talking about a manipulation experiment done by Arif Hamid a few years ago. And here he's just infected VTA dopamine cells with channel rhodopsin, so he can then stimulate them and observe dopamine release in the forebrain. And this here, he's using voltammetry to see this release. And he's giving a half-second pulse train here to give just a brief pulse of dopamine release in the forebrain. And I should say he's calibrated the power and other parameters of the laser to make sure that the amount of dopamine released is somewhat comparable to a moderately unexpected reward. So Arif has actually given these kinds of stimulations in many times within this task. I'm just going to talk about 2 times right now. So one is, if you give the laser extra dopamine right at the beginning of the trial, at the same time as the light comes on. Now, if dopamine is actually involved in motivation in this task, and our measure of motivation is the latency, then maybe giving extra dopamine here would immediately make the rat act more motivated, i.e. have a shorter latency right here. The other time I'm going to talk about is testing this other idea of dopamine being involved in learning. So if, as the animal makes a choice to the left, or in this case here to the right, just as he enters this side port, we give extra dopamine. Does dopamine serve as a reinforcer here? In other words, on the next and subsequent trials, is the rat more likely to make that right choice and not the left choice? All right, so let's talk about that first. So indeed, if we give extra dopamine as the rat completes his choice, then on next trial, he's more likely to make that same choice again, regardless of whether he actually got a reward or not. And this doesn't happen in litter-mate controls. And if we, instead of stimulating dopamine, we suppress dopamine, we get the opposite effect. So this is consistent with this reinforcing action of dopamine in this task. But now looking at the beginning of the trial, just giving dopamine right as the light comes on, what we find is that extra dopamine immediately makes the rat act more motivated. So this is not a learning-driven effect, that requires updating of values that you would then see on the next and subsequent trials. This is happening right away. And you can see as a sort of pulse of increased likelihood of deciding to initiate an approach behavior. All right, so just to say that again, if the rat hasn't already decided to work at the time of this pulse coming on, then extra dopamine makes the rat more likely to decide that engaging in this task is worthwhile. All right. So dopamine can affect both motivation, that is current behavior, and learning future behavior in this task. And it's already not something you would predict just from an RPE only account of dopamine, because our point of RPE is to serve as a learning signal. So now we want to relate these phenomena we see through manipulations to the natural dynamics of dopamine during this task. All right, so there are many ways that you can look at dopamine. We've done a lot of them, not all of them. With Bob Kennedy, we did some somewhat fancy microdialysis. He has some nice techniques where you can measure lots of different neurochemicals simultaneously, about 20, not just dopamine. And you can do that with 1 minute time resolution, which is microdialysis, although no, it's not that fast. So that's going to be our measure, in effect, of tonic dopamine. And then starting with Burgeno, we did voltammetry to look at phasic or faster changes. This is focused on dopamine, much faster time resolution, but has its downsides as well. It's only stable for a minute or two at a time. Most recently, working with Lin Tian and Tommaso Patriarchi, we've been using D-Light, and I'm going to show data from that as well. Okay, so talking first about some microdialysis data. We surveyed, so this is work by Jeff Pettibone in my lab, and Jeff surveyed a number of regions in the medial wall of cortex and medial striatum, and he found that there were certain places in the core, particularly in the core of the nucleus accumbens, where there was a nice correlation between the reward rate that the rat was receiving and the dopamine measure with microdialysis. So that's in the core. If you went more dorsally in dorsal medial striatum, you did not see this relationship, nor did you see it if you went more ventrally or medially into the accumbens shell. So this is like a hotspot. And somewhat to our surprise, we saw a corresponding hotspot in the ventral prelimbic cortex as well. And this is pretty specific to dopamine. Lots of these other things that are being measured are not showing this correlation. This is just showing the same data another way. This hotspot in the accumbens core and then ventral prelimbic cortex. And just as an aside, one thing that I don't know how seriously to take, but I quite like, is if you do a meta-analysis of human fMRI studies and say, where do you see activation that consistently correlates with subjective value? Then you see two spots. So one is in ventral prefrontal cortex here, and the other is in the nucleus accumbens. All right, so now we're measuring the signal with microdialysis. And we can see as we have a block transition, we go from a relatively high reward block to a relatively low reward block, then latencies get more sluggish, right? Conversely, if we go from low reward to high reward, latencies speed up. And at the same time, the accumbens core dopamine is doing the exact opposite. So as latencies are getting longer, dopamine's getting lower and vice versa. So this seems consistent with this Nivdor-Dayan idea of a dopamine reward rate signal that evolves slowly and is used to decide how hard to work. But if still, of course, we're still just measuring something slowly. So is this really a slow tonic motivational signal that's distinct to the fast changes that we see? So of course, now we turn to a faster measure. This is just examples of purple splotches representing dopamine in a voltammetry recording, fast scan cyclic voltammetry. And here's an example of a single session, all the trials, aligned on various events within the trial. So the light comes on, and the rat runs over to the center port to begin the trial. And you see that just before that, you're seeing a quick increase in dopamine starting here. And then when the rat finds out he's going to get a reward in this trial by this reward cue, then you see a rapid increase. And if he doesn't hear the click, the dopamine rapidly decreases. But actually, the largest increases we saw in this task with dopamine were as the rat was running over to collect the sugar pellet on rewarded trials. And then once the rat gets the sugar pellet, dopamine falls fast. Okay. And we weren't by any means the first to see this kind of ramping up with dopamine. So this is now very well-known work by Howell, Paul Phillips, and Grabiel, showing that rats running along a maze arm, you see this progressive increase ramping up as they get closer to the reward. And in fact, even going back to some of the earlier fast scan cyclic voltammetry studies, Paul Phillips reporting that as a rat presses the lever for cocaine, just before lever pressing, dopamine's ramping up very quickly. Another study, Mitch Reutemann and others, showing ramping up very quickly before lever pressing for food, even if there was no cue immediately preceding this. Another more recent example from Kate Wassam and colleagues. All right, so how should we, sorry?
2 minutes.
Okay. So how should we interpret these ramps? So we proposed as a first pass that we should interpret them as a value function. And what do we mean by that? Well, this is simply saying that nearby rewards are more valuable than far away rewards. And so if there's a reward here, you have to pass through many states to get there, then the value of being you, the state value, is increasing as you get closer to it. So it's a natural way of thinking about a ramp. I'm going to skip over this. And we showed in that 2016 paper that the instantaneous dopamine was better correlated with state values than with reward prediction errors alone. There's a problem. The dopamine release rapidly ramps up with reward approach, but in general, this hasn't been seen for dopamine cell firing. So why not? Is it because firing and release generally have not been examined in the same situation? Is it because we haven't looked carefully enough? Maybe recent work by Ochida, Ilana Witten, and so forth suggests that. Are there other methodological difficulties? Or is there a real dissociation between firing and release? And so Ali Mohabi in my lab recorded from optogenetically identified dopamine cells in the VTA in this task. And these blue circles represent the identified dopamine cells, purple are presumed non-dopamine cells, and so forth. And I just wanted to quickly say something about tonic dopamine. So there's been a presumption in the field for many years that there's a thing called tonic dopamine release in the forebrain that's a reflection of tonic dopamine firing in the midbrain, either the tonic firing rate or the number of active dopamine cells. But the evidence for this has always been very indirect. And what Ali found was that if he looked at the firing of his dopamine cells in this task, so first of all, here's the reward rate in black going up and down, gets very low here, correspondingly the latencies get very long, that dopamine cells don't care at all about this reward rate, even though the animal certainly does. And in fact, none of the VTA neurons that Ali recorded, either dopamine, non-dopamine, unidentified, cared at all about this difference in motivation. And so this, we hear a lot about heterogeneity of dopamine cell firing. But this is a case where none of the cells care about this important factor, even though this is directly correlated with robust changes in dopamine release in the forebrain. All right. And this is also true even for a small number of cells that Ali was able to record that were retrogradely labeled from the nucleus accumbens. Okay, so what did the dopamine cells care about? Well, firstly, they care about these sensory cue onsets. So here's the light cue coming on. Here's the go cue. Here's the reward cue, the click. They care about that. But what you don't see is sort of ramping up as the animal gets closer to the food port, or much ramping up as the animal gets close to centering. So whether we look on these slower timescales comparing microdialysis to tonic firing, or these faster timescales comparing firing of dopamine cells to voltammetry, we don't seem to see the same thing, all right? But we wanted more data, or maybe our reviewers did. So we turned to D-Light. We've already heard a lot about D-Light. It's a wonderful tool. And Ali recorded photometry signals of D-Light in the accumbens core during this task. And what you see, again, is first of all, if we bin the D-Light on the same 1 minute bins that we used for microdialysis, then we see the same cross correlation between reward rate and the D-Light signal that we saw with microdialysis. So that's kind of reassuring. But of course, the great advantage with the D-Light sensors, now we can zoom in. And so what happens if we do? So, I mean, these would be enormous 1 minute bins, but of course, the dynamics of dopamine are much faster within here, right? So we don't see the sort of much evidence for slow tonic changes in dopamine. What we see is fast within trial fluctuations that ramp up to rewards. And I'll talk more about that in a second. And if we compare this to firing, so it's certainly the case that these events that cause sharp bursts of dopamine cell firing also cause sharp increases in dopamine release. But at the level of release, we see ramps up, quick, short, sub-second timescale ramps here, that there's no evidence for occurring in firing. This is easier to see if we look at subsets of trials where events are further from each other. So if we consider trials where the latency was more than 2 seconds, so here's where the light comes on, the slow on these 2 second greater latencies, there's not much change in dopamine cell firing. And there's definitely no increase here as the animal sticks its nose in the center hole. But that time is associated with a marked fast ramp in dopamine happening here. And we see the same thing with D-Light or voltammetry. It's just a quicker sensor here. And you can even break it up further by reward rate. So here's the same event, and the VTA firing is seeing not much, maybe a slight dip in firing of the VTA cells, dopamine cells. But the release of dopamine in the accumbens is showing this fast ramp up here. Fast ramp up, fast ramp. So, to some, and this also scales with the background reward rate. So this has proven somewhat controversial because many people have been arguing that, well, maybe there's some firing here that causes this that's just not, you can't just look at average firing rate because there's all kinds of short-term plasticity processes and so forth. We can't find any other property of firing in this period here that changes in interspike interval distributions or anything that would explain this, but we're happy to try out other analyses if people have suggestions. Okay, and when the animal actually hits the reward cue, you see a very nice reward prediction error at the level of self-firing. So you see it's higher response to the same cue if you had a lower expectation of reward. That's a nice positive reward prediction error. And you can see that manifested in very fast dynamics of dopamine release in the accumbens. That's good. So this is a real thing. But for other parts of the trial, what you're seeing is the dependence on reward rate that's just not there for firing of dopamine cells. Okay, and one quick thing I wanted to point out about this, because my friend Uchida has been suggesting that maybe these ramps up that we're seeing here are actually reward prediction errors. And I don't think that's true for a couple of reasons. One is, I'm not sure it makes sense to have a reward prediction error get bigger and bigger as the situation gets more expected. So let me just talk about that. So here's 3 trials with no reward in a row, and then a sequence of rewarded trials. So at the first time the animal finds out it's getting a reward, you see a very nice spike in the delight signal here. That correlates with the reward prediction error of a model. Here, another semi-Markov decision process model. But as the reward gets progressively more anticipated, this reward prediction error gets smaller, and this fast transient at the beginning gets smaller. But instead, what you get is more and more and more ramping up within the trial, and that correlates with the reward expectation at these different times. So given the evidence I presented that dopamine firing is not driving all of these motivation-related dynamics of dopamine release, what is? And we've already heard a number of fantastic talks today, including from Steph Cragg and Pascal Kaiser, talking about the importance of cholinergic control of dopamine release. And indeed, the cholinergic network and the dopamine network are deeply dense and intertwined within striatum. And we've known for a number of years now that if you synchronously activate cholinergic interneurons, you can get striatal dopamine release. But we wanted to know if this was true not just in a slice, but a freely behaving animal. And so Ali took advantage of this wonderful new redshifted version of D-Light that Lynn and Tomasa made and recorded dopamine fluctuations through the same optic fiber that he's manipulating cholinergic interneurons with. And you can see these are spontaneous dopamine fluctuations. And then Ali starts to tickle the cholinergics. You're seeing an increase here, or tickling them for longer, this increase. This is many trials here. So yes, even in freely moving animals, stimulation of cholinergics causes rapid and strong changes in dopamine release. And so the next obvious question is, can these cholinergics be responsible for these motivation-related changes in dopamine release in our task? And so once again, this is what we're trying to account for, the fact that we see the increases in dopamine without increases in VTA self-firing. And if Ali looks at GCAMP signals in the cholinergic interneurons in the accumbens, what he sees is these increases happening at just the right times to potentially drive these dopamine increases. All right. So this is part of mounting evidence that cholinergic neurons have this incredibly important role driving dopamine release, and maybe they're doing that during motivated approach behaviors as well. All right, so to conclude, so I talked about motivation versus learning. So the accumbens dopamine conveys both fast value signals and fast learning signals, tonic and phasic. So I'm not really aware of convincing evidence that there is such a thing as an inherently slow or tonic dopamine signal. Certainly, if you measure things slowly, they can look slow. Or if you're looking at relationship between dopamine and a behavior, like a long approach, and that behavior is slow, then the dopamine can look slow too. But I haven't seen evidence that it's inherently slow. And firing versus release. So VTA spiking does not seem to account for the value signals we see in accumbens dopamine. Instead, these may be sculpted locally by cholinergic interneurons. And I want to thank these folks again, and thank you for your attention.
Well, thanks very much, Josh. We're down to only about 3 minutes for questions. So we should transition right to whatever questions we can deal with.
All right, so I've got one quick one to start with. Is motivation scaling with the value encoded by dopamine?
So yes, so at the same situations that we see the animal more motivated to play the game, and the dopamine is higher. So I think that if that's what the question means, then yes.
And another quick one, do the increases in dopamine in the PL and the accumbens core occur simultaneously?
Yes, they do. So, we did do some simultaneous recordings, and we did look at sort of cross-correlograms between these two things. They're not the same signal, so they both have their independent dynamics, but they both show this correlation to reward rate.
Okay, and one more. So animals are often placed on food restriction to increase motivation for behavior. Is there evidence that food restriction and or non-food restriction, oh, sorry, everything just shifted. Non-food restricted animals don't have changes in tonic VTA firing. That's from Barbara Juarez.
Yeah, so this is actually something we're working on right now, is trying to understand the relationship between tonic firing and states of deprivation and the expectation. And I think that it hasn't been done properly. We're trying to do it now, properly meaning tagged cells and so forth. But right now we don't have any reason to think that being more hungry increases your tonic firing rate.
All right, I think that's all we've got time for, but I'm gonna post the rest of the questions in the Slack channel if you can have time to answer them there.
Thank you so much.
I would like to thank again all the speakers in this session. This is really exciting.
Thank you.